Good morning. Welcome back to FLUOROS Global 2021. This is day 3, if I'm counting correctly, to go through a few administrative slides, but it's great to hear some chatter, same as yesterday, the poster session. It was just nice to have the feeling of some normality again, of real interactions with real people, and of course it's just as nice to be in Hoover and talking to people virtually. So for those here, know your fire exit, that hasn't changed. Behave responsibly, no question. There's recordings, there's copyright. Speakers' views are their own, they don't respect the funding agencies. Do everything for Hoover, particularly for the posters. Have the masks in this physical setting. Maybe tonight for the drinks we can actually venture outside. The rain seems to have let up. Land acknowledgement. We still gather on traditional lands of the Mashpee, Niantic, and Narragansett tribal nations. We honor their histories, ancestors, and perseverance in our communities today, as our country was built on indigenous land. We acknowledge the indigenous people and honor their stewardship of the land, sea, and resources that they hold sacred, and that we shepherd together. And already time to say thanks to the Zurich group, Zanjun, Martin, Valentina, Lena, Jona, Anna, and Ian, who did a fabulous session through Hoover. It didn't feel that remote, it worked really well, and of course on the local side, Yitka, Emily, and Alicia, who worked behind or in front of the scenes to make this all working. Same to the Beijing group. With some initial technical trouble, we got a nice set of talks. And I should point out a big thank you to High Output in the Inner Space Center. Those are the folks that you never see, who run everything behind the streams. And some of you probably realized that yesterday Facebook and WhatsApp and some other things went down, and what we used to stream also went down. So there was a bit of a panic for a little bit. I mean, not panic, there was just a realization we had an issue, but luckily during the lunch break everything was solved and we could continue streaming. So to the outside nothing ever happened. Behind the scenes, big thanks that you worked to make this work. And then of course Waterfire and Russ Morantz for being very flexible with what we're doing here. So thank you very much for the good support. All right, and I should say this has been a long time in the making. Today really is Judgment Day, at least for the 30-odd poster judges. Please enter your scores by the end of today, US midnight, so we can select those winners for Thursday's acknowledgements and their prizes. So with that, I say thank you, and I introduce Anna Robach. She's a postdoc at Mount Sinai, and she'll guide us through the morning session. Anna, the podium will be yours.
Okay, excellent. Reiner, advance with the green button. Excellent. Okay, well, get oriented here. I'm kind of a walking Apple ecosystem. I have all my notes on my phone, my iPad, but I think we are ready to go here. So Reiner, thank you so much for the introduction. As many of you know, this time last year I was still Reiner's PhD student, so it's just terribly exciting to have him introduce me as a postdoc still. I can certainly get used to that, but anyways, good morning, good afternoon, and good evening. Thank you so much for joining today's programming. I'll be the master of ceremonies for today, and I am so excited to share today's events with you all. As you may see in your programs and agendas, up first today we have a session discussing the bioaccumulation and ecotoxicology of Pfas, chaired by Dr. Amila De Silva from Environment and Climate Change Canada. We'll have a break for lunch, and from there we'll move into a pre-recorded session from Australian colleagues focused on remediation, chaired by Dr. Joachim Mueller. However, before we kind of move into the events for today, I was given the chance to frame today's topic before we start with the technical talks for today. Like any good postdoc, I eagerly accepted the opportunity to get in front of a crowd of my peers, but I was more importantly more excited to kind of take this opportunity to comment on where we are and where we're going in terms of bioaccumulation and ecotoxicity. Oh, there we go. Now I got it. Great. So as it turns out, as I started thinking about where we are, looking at the literature, it's clear we've made incredible progress gathering information and building expertise around this topic over the last 2 or 3 decades. On the slide here, I have some figures from one of those first papers that showed us PFAS, and particularly PFOS, really are global contaminants. This work came out something around 20 or so years ago with this study as well as other studies that documented the presence of PFOS in seabirds, fish, reptiles, and amphibians worldwide. Other contributions around this time further established the presence of this compound and other legacy PFAS in specific wildlife species at specific locations, including key work documenting legacy PFAS in Arctic wildlife, Great Lakes wildlife, and the transfer of these compounds in food webs. That initial handful of studies has swelled to hundreds of efforts that have rapidly expanded our knowledge on PFAS bioaccumulation and ecotoxicology concerns. We are now aware of thousands of different compounds that are considered to be PFAS and had found diverse formulations beyond the perfluoroalkyl acids are capable of accumulating in wildlife all across the globe. For example, work led by our first sessions chair today documented the accumulation of perfluoroalkyl phosphinic acids in fish, birds, and dolphins from across North America. The width and breadth of our investigations has just drastically expanded to include a wide array of species and habitats, including commercially relevant species, threatened and endangered wildlife, remote wildlife, wildlife adjacent to contaminated sites, and varied trophic levels ranging from planktonic consumers to large whales. On the slide, I've put up a few images of some of the fish and wildlife species that just happen to have some of the more photogenic Wikipedia photos, but this is purely a quick summary in that if we were to put up photos of everything where we found, or excuse me, every species where we found PFAS, we'd be looking at hundreds and hundreds of slides, unfortunately. In tandem with that, we've gained some key insight about acute and chronic toxicity of particularly legacy PFAS in different taxa ranging from model organisms to observational studies in wild species. Unfortunately, found varied negative impacts across these taxa and life stages. And so despite this accumulation of a lot of information over the past 20 to 30 years, we still possess far too little information regarding, sorry about that. We still possess far too little information regarding the totality of PFAS that occur in wild systems and how PFAS may be associated with acute and chronic impacts across the diversity of wildlife in which they are found. On the slide here, I tried to brainstorm things that I felt confident in as someone who's been working on PFAS now for quite some time. But I quickly found out there were way more questions and topics that we feel less confident about. These uncertainties and questions are numerous and multifaceted, and I'm certain that my little list here can be readily augmented by the expertise and concerns of researchers and community groups throughout this program. So our work is definitely cut out for us as a research and activist community as we continue to explore bioaccumulation and ecotoxicology questions. And yet, I want to assert that as we tackle these key data gaps and questions, it's important to remember that focused research in wildlife helped answer questions from the outset of our exploration of this topic. The questions that I now sort of consider the tip of the iceberg here, things that we think are apparent, were once totally opaque. As we continue to study these compounds, it's key to continue to leverage the utility of wildlife as indicators of our shared global environment. So for example, some excellent work out of the Canadian Arctic, like this study and others from the early 2000s, demonstrated the long-range transport capacity of these compounds and the biomagnification ability of PFOS, where we see that PFOS increases with trophic level in an Arctic food web consisting of plankton, fish, and marine mammals. The biomagnification in this example occurred in ways that we hadn't seen yet to this point when we're looking at PCBs or other hydrophobic organic contaminants. And so as we look ahead to today's challenges, while we may feel confident in how PFOS biomagnifies, for example, well-designed wildlife studies have the same capacity to tell us about the evolving nature of PFAS in the environment. As several talks in the program so far have alluded to, we have a lot to learn about unidentified PFAS circulating in our environment and in our bodies. We quantify these unknowns, and we're increasingly learning how to do so using organofluorine measurements, or EOF measurements, as we've heard. And thus far, the application of this technique has let us know that different trophic levels are associated with different proportions of this unidentified organofluorine. For example, on the slide here, I have some values collected by Dr. Anna Sharman's group that showed the percentage of total organofluorine accounted for by targeted PFAS, the numeric values on the slide, that changed by species and trophic level, with the most unidentified organofluorine in water and lower trophic level aquatic matrices. Likewise, excellent work led by Kira Spahn measured target, non-target, and bulk organofluorine in a number of marine mammals from across the North Atlantic. That work pointed out some key geographic differences that we can follow up to explore on, namely marine mammals from the coast of North America. Oh, thank you, Reiner. Always saving the day, per usual. So now we're on this slide. Rewind just a little bit. I'm not going to repeat myself, but these EOF measurements in wildlife are pointing us towards questions we need to continue to ask across the diversity of ecosystems and within our own populations and bodies. The same idea applies as we try to grasp trends over time in the environment. Wildlife studies in the past have provided important commentary on the evolving nature of PFAS production, use, global transport, and accumulation. So as mentioned in Dr. Elsie Sunderland's keynote talk on Sunday night, work out of her group previously documented declines in Phosa and Pfas in pilot whales in the North Atlantic. Those same trends showed up in human populations when we looked at that. We see the same trends reflected here in this study led by Holly Barrett, which found similar declines in Pfas and Phosa in belugas from the St. Lawrence estuary over the early 2000s and into the 2010s. As well, that same work pointed out increasing concentrations of emerging short-chain chemistries, replacement compounds, and fluorotelomere carboxylic acids. Likewise, in my own work, wildlife gives us insights into trends that we're not going to find necessarily looking at water or even some of our own current blood chemistry. In my dissertation, we measured the ether sulfonic acid Naphion byproduct 2 in a sample set of seabirds, great shearwaters, dating back to 2010. As it turns out, we found that novel PFAS in birds from the offshore Atlantic Ocean dating back at least to 2010. As you may remember, that compound hadn't really been identified yet, even in the area it was being produced, until work put out in 2015, meaning the compound was already circulating widely in the environment before we even knew it was there. And so while I hope I've given you a big picture glimpse of what we know about PFAS bioaccumulation and the continued utility and imperatives that we have to work with as we move forward, the future of this work is also so key because humans are directly exposed to PFAS through abundant consumption of animal products, wildlife, and fish. Some of my own research really speaks to this poignantly for me, and I wanted to share that here with you today. I'm from Philadelphia, Pennsylvania, and previous exploratory work in this region has demonstrated the presence of a series of novel chlorine-containing PFAS in soil, surface water, and groundwater from southwest New Jersey and the Delaware River and estuary. These studies showed that these chlorinated species were present in soil all the way up to New Hampshire and were found in groundwater and surface water wells around a particular suspected source. Over the course of finishing my dissertation, I got really curious about what this meant for local wildlife. I sampled fish in tandem with recreational fishermen across a 50-mile expanse of the Delaware River, and unfortunately found high levels of these novel chlorine-containing PFAS in every type of fish that was sampled from this river, at concentrations up to around 6,000 nanograms per gram PFAS in these fish. What really speaks to me, though, is when I look at these data points, something that Emily Donovan pointed out, behind all of these data points were people. Here are my people behind those data points. On the left is a gentleman named Rob who went above and beyond across the scope of my study to get me fish at all of these different locations that you saw on the previous map. But the thing that really gets me and kind of chokes me up talking about this, Rob would give me the small fish and take the big ones home to his family and friends so that they could have a special Sunday meal. On the right-hand side, that's my dad. I grew up in this region, and after work, my dad would take us fishing, and occasionally we'd get a catfish and bring that home as something to eat from this area. So behind all of these data points that we're gathering about bioaccumulation and toxicology, there are people and communities who need this information. So with that, I am going to conclude and just summarize that as we head into today's sessions, I challenge us to celebrate the foundations that we have built while preparing us for PFAS challenges in the future as we continue to explore this topic in wild and built systems. How can we design and implement wildlife and ecotoxicology studies to backfill our knowledge gaps while pivoting to address new research questions, new PFAS, new species, and new toxicity endpoints, and so on? And so with that, without further ado, I'd like to stop my introduction here and move to introduce the session chair for today's Bioaccumulation and Ecotoxicology study. Dr. Amila De Silva is a research scientist at Environment and Climate Change Canada and the Canadian federal government. Amila has built a research program there on the fate and transport and bioaccumulation of priority organic contaminants, often using advanced analytical instrumentation. She has studied PFAS throughout her professional career, beginning in 2002 during her PhD studies in environmental chemistry at the University of Toronto. In her current position, Amila has tracked PFAS in the Arctic, including environmental matrices like glaciers and permafrost, as well as wildlife like beluga and seals. Closer to home, Amila has studied novel PFAS in the Great Lakes region, in diffuse and urban sources, as well as in wastewater, fish, and aquatic life. Amila is a first-generation Canadian and lives in Burlington, Ontario. Please enjoy today's questions, and I will turn things over to Amila.
Hi, everyone. Greetings from Burlington, Ontario. Thank you very much for that introductory talk, Anna. I have pre-recorded my 15-minute introduction to this session where I will be introducing the speakers and really the first decade of PFAS research and what we learned specifically in Canada. So without further ado, Alex. Welcome to this session on ecotoxicology and bioaccumulation of PFAS. I'm Amila De Silva, and I'm a researcher for Environment and Climate Change Canada in the Government of Canada. Our session aims to cover everything from microorganisms to the apex predators such as polar bears. So this is a map of Canada. I find this depiction somewhat humorous because it's as if Canada is an island. However, in fact, Canada is just another country on this planet. And since the earliest discoveries of PFAS, it was clear that PFAS are a global contaminant. It was with this in mind that the first ever FLUOROS meeting was launched in the year 2005 in Toronto, Canada. I was a graduate student at the University of Toronto at that time, and all of Scott's students were part of the local organizing committee. Yep, that's right, that's me in the first row, left corner. You can see the byline for FLUOROS contains the word international, and this was to emphasize the international aspect of PFAS contamination on a global scale. So why was the first FLUOROS meeting an international meeting initiated and hosted in Toronto, Canada? Well, I think it probably had something to do with the intense focus and research on PFAS coming out of Canada at the time. This paper in 2001 was published by John Giese and Kanan, and it was the first environmental distribution of PFOS, this molecule shown down here, in wildlife collected from across the world. It would be another 8 years before PFAS was added to the Stockholm Convention on Persistent Organic Pollutants. but a few years earlier to that, Canada proposed virtual elimination of PFOS, and this was largely due to research coming out of Canada during this decade. Less than a year after the Giese and Kanan study on PFOS, Scott Maybury and his group published this paper. This was the study of PFAS in a creek downstream of the Toronto Pearson International Airport. In this creek, in fish collected here, Scott and his team identified PFOS, just like Giese and Kanan, but also this long suite of perfluorocarboxylic acids from the C7 version right up to the C14. This is the C14 perfluorocarboxylic acid shown here, and just look at the number of fluorine atoms. Now, the discovery of this sampling effort was really driven by this equipment malfunction at the Toronto Pearson Airport. But what was interesting is that Scott and his team also found these contaminants in the upstream reference location. So this was our first inkling that perfluorinated acids, including PFOS and others, were a ubiquitous phenomenon. I joined Scott's group as a PhD student a few months after this paper came out, and this began my career in the analysis of perfluoroalkyl substances. So the 2001 discovery of long-chain PFCAs in fish really triggered an important research question. Why? There is no indication that long-chain PFCAs were an important industrial product. PFOS, of course, was known to be associated with AFFF, and PFOA was known to be a fluoropolymer processing aid in the manufacture of Teflon. There wasn't any commercial relevance for these long-chain PFCAs, so how did they get there? This decade of PFAS research really looked into the why question and highlighted the role of precursors. So this figure here represents a hypothesis from Scott's group of how long-chain PFCA could have such a ubiquitous ecological presence. Scott and Joyce Stinglassen hypothesized that commercial products that we use in surface treatments, such as treating your couch and your rug, textiles and garments, that contain PFAS could release these labile fluorocontaminants. And they tested this. They purchased these products off the shelf and sampled the air above the liquid and identified these fluorotelomere alcohols. The one that I show up here is the 8,2-fluorotelomere alcohol, also known as Phos. But in Joyce's paper, they actually identified the C6, 8, 10, and 12 version of this substance. Around the same time, studies were reporting the widespread presence of Phos and other precursors in indoor and outdoor air, such as this paper down here by my colleague Mahiba Schweib from Environment Canada, and Naomi Stock, who did an air sampling campaign in the North American troposphere. Like PFCAs, these Phos were proving to be a ubiquitous PFAS contaminant. But what was the link between Phto and PFCAs? Well, you can see that the hypothesis is really based on the similarity in the chemical nature of these two substances. This is PFOA and this is Phto, and they both have this long perfluoroalkyl component to them. So Joyce Stinglassen did a biodegradation study with microbes and found that when she incubated 8,2-Phto with microbes, the biological degradation product was this substance. So this was the first link between precursors and persistent byproducts. We'll hear later on in this session from Jinxia Liu, who also conducted biodegradation studies of precursors. Abiotic pathways were also important. So this study by David Ellis and John Martin, who were postdocs with Scott at the same time, went down to Dearborn, Michigan with 8,2-fluorotelomere alcohol and partnered up with scientists from the Ford Motor Company, Tim Wallington. And they inserted 8,2-Phto into this atmospheric chamber and simulated the atmosphere and collected the byproducts and found that 8,2-fluorotelomere alcohol would also produce perfluoroalkyl acids through atmospheric oxidation. So we now have these two different pathways biological transformation in microbes and mammals, and including humans, but also just in the atmosphere. And all of this research was being generated in Canada at the time. So I started off with this map of Canada in the global context. Canada is also part of the circumpolar Arctic. So if we take this view where we're looking down at the top of the globe, this little plus sign is roughly the location of the North Pole. You can see the interconnectedness of Canada with other nations that occupy the Arctic Circle. Towards the last half of the 2000 to 2010 decade, big discoveries were made of PFAS in the Arctic. Again, another breathtaking study by Mahiva Schweib and Tom Harner from Environment Canada, where they looked at PFAS, including fluorotelmer alcohols and polyfluoroalkyl sulfonamides, in air from Greenland to Norway and Iceland, the Faroe Islands, and throughout the Arctic archipelago of Canada, and found these substances. Nobu Yamashita from Japan looked in the ocean and found a global survey of perfluorinated acids in all of the oceans. So together, these two papers are showing different long-range transport mechanisms that could result in perfluorinated substances in remote areas such as the Arctic. Accumulation of PFAS in Arctic wildlife was confirmed by one of my favorite of all-time papers, which happened to be by John Martin and, of course, world-renowned Arctic scientist Derek Muir. They looked at both marine and terrestrial organisms in the high Arctic of Canada and found high concentrations of perfluoroalkyl substances. This was the first discovery of long-chain perfluorinated acids in Arctic wildlife. So clearly this was a very exciting time of PFAS discovery from 2000 to 2010 in our understanding of precursors, mechanisms, and global transport of PFAS, and ultimately the accumulation in wildlife. In the 10 years that followed, I saw huge changes in the PFAS research network, greater research being done in other geographic areas, discoveries of polymers, precursors, lots of studies on AFFF, human exposure through food, drinking water, and products, and great innovation in toxicity studies. So this leads me to where we are today. It's 2021, and we are still researching and discovering PFAS. In our session, we will present a comprehensive current snapshot of the ecotoxicology, bioaccumulation, and wildlife exposure to PFAS. I approached Rob Letcher to kick off this session with a keynote talk on PFAS and wildlife. Rob is a colleague of mine at Environment Canada. And when I first met Rob, I thought of him as the polar bear guy, because he was putting out these very unique studies of contaminants in polar bears from Canada and Greenland. But actually, I learned that Rob does a lot more than polar bears. He's worked extensively with birds and fish, not just in the Arctic, but also in the Great Lakes. He has many, many years of expertise in environmental chemistry of organic contaminants, including brominated and chlorinated pollutants. This extensive and diverse expertise really shows in the quality and impact of his research program. So I'm really pleased that Rob has agreed to speak in our session today. Now, I mentioned Rob will be speaking about bioaccumulation and biomagnification of PFAS, but I wanted to bring in another expert to talk about what we can learn from field and lab tests in bioaccumulation. Pierre Labadie immediately came to mind. Pierre is a researcher in southwestern France at CNRS, the National Scientific Research Center there. And what really caught my eye is Pierre's experience in the marine and freshwater environment, as well as estuaries and terrestrial organisms also. Pierre has done studies of contaminants in these major rivers in France, the Seine, the Rhône, and the Garonne. I also appreciate that Pierre's targeting novel PFAS. Probably one of the loftiest responsibilities in this session would be the coverage of ecotoxicology studies. Ecotoxicology of PFAS is such a broad topic with so many areas that one could possibly cover. I'm really confident that Christopher Solis has the ability to deliver on this. Chris is a professor at Towson University in Maryland, and he's really plugged into different perspectives that probably come from originally being a research toxicology for the US Army, and then an ecological risk assessor for the US EPA, followed by 6 years at Texas Tech. Chris has been doing PFAS ecotoxicology experiments in many different types of organisms, from these freshwater midges right up to avian reproductive toxicity with quail. Chris has also worked with snails, frogs, reptiles, oysters, fish, mosquitoes. I might be missing a few. So he really is an expert in these ecotox studies from very different ecological niches. He was also a co-author on Jerry Ankley's monster review paper on ecological risks of PFAS that was published last year. That final printed copy was almost 35 pages. So like I said, I'm pretty sure that Chris can summarize the key points of PFAS ecotoxicology in a 20-minute presentation. I want to thank the speakers and the organizers, and as well as Steep Funding, for this meeting. I really hope you enjoy this session, and I look forward to a lively discussion during the question period. Thanks. Okay, great. That was a very good presentation, Emilia de Silva. So first off, we are going to have Dr. Robert Letcher, who is a research scientist from Environment and Climate Change Canada, located in Ottawa, Ontario, and Rob is also an adjunct professor at Carleton University. Rob will be delivering a keynote address on bioaccumulation, wildlife exposure of PFAS. Thank you, Rob.
My name is Robert Letcher. I'm with Environment and Climate Change Canada, and I'm happy to be a keynote speaker in the session today on microorganisms to polar bears, ecotoxicology, and bioaccumulation of PFAS. So what I'm going to give you today is an overview of PFAS in wildlife and to some degree in fish, but with a focus on the Arctic and northern regions. First, a brief outline of the presentation today. Basically, we'll start with discussing PFAS in general. How many are there? Which ones are of elevated concern in biota, particularly to wildlife? Talk about regulatory activity for PFAS. For example, Uneps Stockholm Convention on POPs and new replacements. And then we'll talk about the Arctic, major programs that monitor PFAS in Canada and internationally, and as a source of PFAS information in wildlife. The bulk of this presentation will be a historical review of published PFAS literature in a global context up until 2017 and up until recently, and with a focus again on wildlife in the Arctic. And then post 2017, I'll discuss a few new cases, studies of PFAS in Arctic wildlife and fish, and then have a wrap up with summary and some take-home messages and thoughts. So, of course, all of you are familiar with PFAS, and you've probably heard some of this information before, but just a little context check that PFAS are found in an incredible number of products. They are comprised of a very large family of different chemical groups, and this was or has been exemplified by a recent paper by Buck et al., where you can see it off on the right here, where they talked about the identification and classification of commercially relevant PFAS, and basically found there to be about 4,700 PFAS listed in a 2018 OECD UNEP report that were directly connected to commercial products based on input from the three major producers. And in the environmental context, I put in a rectangular box down in the middle bottom, just showing how many of these compounds are in fact carbonyl, sulfonyl, or phosphate acid type compounds. And it's these perfluoroalkyl acids that we refer to as the forever chemicals. And the focus of much research from the very beginning was on PFOS, so the C8 sulfonic acid, PFOA, the C8 carboxylic acid, and more recently the perfluorohexane sulfonic acid, as you can see to the right. And of course, there are compounds of differing chain lengths, but these compounds are important to know in a wildlife context, and particularly in the Arctic, as they will be shown to be a dominant presence in terms of PFAS. Just a little context. I know Dr. Liu in the session today will talk more about precursors of PFAS and biotransformation, biota, but it is really important to emphasize that they're using PFAS as an example, that there's an incredible amount of pre-FOS, that is compounds that basically can be converted either abiotically or biotically into intermediates that end up, in the case of PFAS, end up as the sulfonamide ultimate precursor to PFAS, as you can see in the middle of this flow diagram. And basically the process to arrive at that forever chemical labeling, as is the case with PFAS. A little bit about regulatory priorities in Canada and are mirrored elsewhere in the world with the, and as you will see, and I'll talk more about it, the regulation of phasing out of C8 type chemistry for poly or perfluoroalkyl acids like PFOS and PFOA. There has been attention shift to shorter chain carboxylic acids, shorter chain and longer chain carboxylic and sulfonic acids, as you can see in the top. And you can see this was from last year. This is basically a table showing basically information available for various types of mainly, well, in fact, totally, as you can see to the far left, perfluoroalkyl acids. You can see the information that exists, where the red means that there's basically no data. And you can see when you look to the far right on concentrations in Canada and water, soil, sediment, biota, effluent, you can see that there's a lot of red and a lot of yellow, which means that there is still quite a data deficiency. And that, again, that's probably mirrored elsewhere in different jurisdictions. Environment Canada has now assessed up to 200 PFAS using a class assessment approach. And of course, these are basically mainly acids and their precursors. Now, you'll see more of this, but there is particularly for PFAS, there is a whole variety of replacements coming online. And this is just a sampling of those replacements. And basically, as you will hear throughout the talk here, there's a very large data deficiency in these compounds. And compounds such as Gen X and Adona, and the perfluoroether sulfonic acids such as F53B are increasingly being analyzed for, but not necessarily detected, as you will hear shortly. So the ultimate assessment, I guess, process for regulation internationally is known as Uneps Stockholm Convention on POPs. And this is a rather busy slide, but it gives you a timeline of, and how long the timeline has been, 20 years to be specific, over which time compounds have been risk assessed and eventually listed under one of the annexes of the convention, where Annex A is elimination. And you can see the letters are off to the right of the different compounds, but you can see it was only in 2009 that the well-known PFAS and its salts and its precursors were put under Annex B of the Stockholm Convention. It was the first such perfluorinated compound or fluorinated compound to be put on the list. And it wasn't until 2019 that PFOA was added, again, with its salts and precursors. And right now there is an assessment going on with a likely listing of perfluorohexane sulfonic acid. But there is a minimal number of compounds that have reached this level of regulatory attention. So over the start until right up to the present, I carried out basically a literature search of the Web of Science to get an idea of what sort of numbers we were talking about in terms of publications that have been made. Don't get hung up on these numbers per se, but just the magnitude of difference. You can see using keywords such as PFAS or PFC in combination with various media like water, air, sediment, biota, wildlife, etc. You can see on the left in the blue that that starting in 2004, there's been thousands of papers published with respect to PFAS or PFCs and water. and to a lesser degree in sediment. But if you look down, farther down the left to in the red there, you can see a very relatively small number has been reported for wildlife. Again, these numbers might not be particularly and totally relevant or reflective of what's in the literature, but the point is that there's a very low number compared to abiotic media. And if you go over to the right, you can see using more traditionally used keywords like perfluorinated compounds or perfluorinated acids, and or just targeting PFAS, you can see that the numbers are in the thousands of papers since the start of reporting, but relatively speaking, we're in the hundreds when we come down to wildlife. So let's go back in time. There has been a number of reviews over time that have helped to kind of frame what PFAS looks like in the environment, in particular in wildlife. And you can see on the far left there was the well-known first paper that reported PFAS in free-ranging biota by Keynote et al. In 2001. So we're basically 2 decades since the first report of PFAS, of any type of PFAS in wildlife. And you can see on the bottom, these are plots that are relative to a review that came out about 5 years later, the first such review on biological monitoring of PFAS, and you can see you get an appreciation on the bottom of the types of information that were summarized. So you have birds from the Baltic, from the USA and Asia, and then you have mammals. This is PFAS, by the way. You can see, again, from Arctic regions, from Europe, and elsewhere in the USA. So basically, the species that had been reported on were mainly fish, birds, and marine mammals from North America, Europe, and Asia. And again, the data was limited to PFAS and largely C8 to C12 perfluorinated carboxylic acids. There was another follow-up review to the 2006 Good et al. Review that looked at the situation up until 2010. And basically, similar reports were coming, although there was a wider range of geographical locations that were reporting PFAS and mainly PFAS, including South America, Russia, Antarctica, and different climatic zones. So an increasing amount of information was coming from the Arctic. Basically, they all were showing, again, elevated PFAS and PFCA levels and an increasing breadth of vertebrates, reptiles, but again, mostly fish, birds, and marine mammals, the predominance of PFAS, and those mid-chain length PFCAs. And most importantly, in terms of replacements, the detection of PFCA and PFSA precursors was not frequent in environmental samples at all. So again, if we look up to 2010, here's a kind of a general kind of collage of studies that came out over that period that were related to mammals, related to wildlife. And you can see that, again, predominantly, there's a diversity of animals, but predominantly you have, again, PFAS and a suite of PFCAs. So in the US, Canada, Greenland, also in Lake Baikal and Central Asia, Russia, Japan, beluga whale, and elsewhere. The same goes for birds. Again, don't take these as exhaustive, but they are very reflective of what was reported between 2006 and 2010. And again, you can see different species of birds in the Baltic, in Iceland, in the Faroe Islands, in Japan, were being reported during that time period. So now, as of 2010, there was a review on PFAS that just focused on the Arctic. And this is a review by Budd et al., as you can see, and Stoughton. And again, you hear the similar theme of findings in these reviews. Basically, a difference in this review is they talk more about the dominant PFAS transport pathways to the Arctic, atmospheric and oceanic, particularly of precursors. But again, biological monitoring in the Arctic focusing on PFCAs and PFSAs, particularly PFAS, very few data on precursors. One difference was there's some report on marine food web and PFAA accumulation. It was in greater study, particularly in top predators. But the majority of temporal and trend studies at that time were, again, North America, Arctic, and Greenland, and showed increasing levels of PFAS from the 70s. And of course, this of course is about the time that PFOS, for example, was added to the Stockholm Convention under Annex B. When we get to, if we hone in more on the Arctic, we have to talk about two very important programs. In Canada, there's the Northern Contaminants Program, which has been going on for more than 25 years. And you can see the species and the different media that have been analyzed over time. And you can see polar bears, top predators in marine environments, caribou and fish. And the information that comes out of the NCP feeds in onto the international level to a program called Arctic Monitoring Assessment Program, or AMAP, under the Arctic Council, just to your left there. And again, these repositories of information are important in feeding into regulatory processes, such as with the Stockholm Convention. The AMAP, as well as the NCP, put out reports every year, and they are quite large in number. And recently, for AMAP, they put out a report on chemicals of emerging Arctic concern. And so at that time, they had found that there was at least one report or measurement on greater than 150 individual and 18 groups of substances in the Arctic. And among those compounds you can see on the right were new and replacement PFAS. With respect to the Arctic, a very recent review came out that was derived from that AMAP report that focused specifically on PFAS in the Arctic by Muir et al. in 2018. And what you can see here, just point out that you have off to the left, you have above the red line all the perfluorinated acids, and below the red line precursors, where there was some report, at least some compartment of media. You can see a lot of those precursors are relative to PFOS, and then you have the fluorotelomere alcohols that would pertain to some of the carboxylic acids, perfluorinated. But you can see that there's an absence of X's below the horizontal red line, and of course you don't see a lot of replacement compounds, a lot of precursors, but not replacements. You also see that there's lesser number of X's, meaning no data for biota, for example. And but you can see that for the acids, there's a fair amount of data for biota. But again, it gives you a general impression of the data situation. So emerging and replacement PFAS, potential Arctic concern. Again, I just show these compounds again because you can see that there has been some detection of some of them in the environment and in Arctic wildlife. You can see in the blue off to the left, compounds like F53B have been detected, and PFEX, which is a direct replacement for PFOS. And there have been compounds that have been analyzed for but not detected. As you can see here, off to the lower right, you can see compounds such as GenX and Adana, for example. So getting into some case studies of newer information, this really came out just after the review by Muir et al. But you can see here, this is one of the first reports that reported on detectable or quantifiable levels of replacement PFAS, such as F53B, as you can see in the left. But you can see, and of course, shorter chain sulfonic acids like perfluorobutane sulfonate in the middle there. But if you look to the left, you can see that's a log scale, and you can see the PFOS still dominates in terms of concentrations, and this is in the liver samples of polar bear from East Greenland, seals, and also killer whale. And this was the first report actually to report on PFAS in killer whales. This study is a study that I've led over these years, and this is a study on circumpolar PFAS and polar bears that is due to be submitted for publication soon. It's a bit dated, but you can see here, this is still the only widespread study that looked at spatial comparisons of PFAS and polar bears from different subpopulations around the Arctic. As you can see on the left, the red circles. On the right, you can see Pfas, again, the dominant compound. And there was some report back in 2005 by Smithwick et al. On Pfas, in particular, in polar bears from several of those populations. And you can see that for the dark blue bars, which are 2006 to 2008 samples, you can see there is a consistent decrease relative to the Smithwick et al. Study back in 1996 and 2002. So some indication of decrease. And of course, we know that PFAS and its C8 chemistry was phased out by the major producers around the early 2000s, 2002. So at that time, considered a good news story. What we also did with this circumpolar study, where we could, we looked at linear and branched PFAS isomers. We know that electrochemical fluorination processes are not clean, and they do produce linear PFAS, which we mostly refer to, but also some branched impurities. And in fact, commercial PFAS is known to be 86% PFAS, where 65% is the linear and 35% is the branch. So there's a fair amount of branched in the commercial mixture. But in polar bears, you can see here off to the far left, linear PFAS, highly dominant in the thousands of parts per billion wet weight. And then, but you can see in the left bottom, you can see then the relatively much lower concentration of the mono and di-branched PFAS. And you can see at the very bottom that in polar bear liver, basically all of the PFAS is linear. But you can see by the plot that there is some differentiation in levels of the mono branched PFAS through polar bears, where the levels are higher in Baffin Bay and in East Greenland relative to other circumpolar populations. Recently, since that 2006-2008 study, we reported on 2013-14 polar bears from Hudson Bay, the two subpopulations, South and Western Hudson Bay. And of course, Hudson Bay being a climate change hotspot with a more well-known understanding of the food web in that marine ecosystem. And we analyzed for about 300 different compounds and detected 210 in liver or fat, mainly fat. But what I want to point out to you here is that the report of levels are consistent with what we saw 8 or 9 years ago. At the bottom right, you can see some of PFCA's 12 different isomers, Pfas, perfluorohexane sulfate, and Phosa. And you can see the levels are still high, and of course, much higher than many of the other compounds that we analyzed for. Trends. So this information, we're now kind of terminating at the end of the 2000s, the trends. And you can see here that PFAS at the top, you see the addition of the Stockholm Convention in 2009, that PFAS is behaving trend-wise differently between the two subpopulations, but the rate of change is not all that great, and certainly not declining since 2009. The same thing goes for the perfluorohexane sulfonate, and then you have the two of the major carboxylic acids at the bottom, and we see some showing trends of no change and some increasing. And if you look to the far bottom right, you can see a paper by Wang et al. Talked about scenarios for global emissions of C4 to C14 PFCAs, and even now those emissions are considered to be high. So this could be the reason why we see no decrease in trends, because of the influx of precursors that can be converted to the carboxylic acid end products. When we think about trends of PFAS and other compounds in polar bears and other wildlife, we have to be aware that there's a lot of confounding factors. You can see biological, abiotic, and ecological factors, climate change being premier among those, and they change the diet and food web of polar bears, for example. You can see, and we know that from stable isotope and fatty acid analysis. So just something to keep in mind in interpreting temporal trends. Again, here's some work for just PFAS that we completed for Western Hudson Bay polar bears, where we looked at a number of climatic variables in statistical relationship to PFAS levels in polar bears over time. And what you want to take away here is that, for example, the total accumulated cover of sea ice shows an increase over time up till 2015. And you can see that the slope has increased for, when we adjust for that and the PFAS trends, it tends to be an accelerated accumulation of PFAS in polar bears. So evidence of climate change affecting trends of a major PFAS. Similar results for polar bears and ring seals from Svalbard, Norway. Again, these are two studies by Rudy et al. That looked at predator prey from this region. And we can see that basically PFAS is still dominant. The mid-length PFCAs still of concern. and of course, the trends, we see some decreasing and some increasing for both. But they more or less reflect one another, at least from this population standpoint. Bioaccumulation. This is a paper we put out on East Greenland polar bears and ring seals a few years ago. But we really wanted to look at the underestimation of accumulation of mainly perfluorinated alkyl acids. We can see, we know that bears eat mainly seal blubber and don't eat liver. And when we adjust the magnification factor on that, you can see how much higher the biomagnification factors are for situations where you can measure the particular PFAA and the seal and the bear. Just a couple other studies. This is a land-based study on PFAS accumulation of fate in Arctic marine food web, right up to birds. This is again Norway. There's a number of studies that have been published out of Norway, and we can see here what's interesting about this is they looked from zooplankton, crab, also an abiotic media. I'm not showing that here, but zooplankton, crab, fish, up to a major predatory bird, the glaucoskull, and you can see along the bottom going to the right the shifting proportion of the PFAS suite, where you have more and more of the longer chain PFCAs coming in, and then by the time you get up to glaucoskull, you have mainly PFAS, of course, and then you have C9 to C11 carboxylic acids. Another one, this is a little bit older, but it's a Faroe Island Greenland study, basically again showing fish, terrestrial mammals, and birds from three locations, and again showing how terrestrial creatures have lower levels and you have higher levels and those associated with marine environments, although there is some differentiation among the PFAAs. You can see here by the C9, C13, carboxylic acid, and PFOS. So to wrap up, some of the highlights, I guess, and takeaway messages from the PFAS in the Arctic wildlife and fish. Again, there is some new data for a wider range of PFAS in the Arctic wildlife biota since Myrdal in 2017. It's only been a few years, and of course, we're in the time of COVID, so the rate of publications coming out has likely slowed a bit. Again, still, after all this time, most data is PFAS, a few PFSAs, shorter chain included, shorter chain and longer chain PFCAs, but basically those shorter chain moieties are not being detected or at very low levels in wildlife and fish. We see extensive data sets for long-term trends of PFAS and other PFSAs, and some of those data sets are using archive samples going back to the 70s and 80s. So up until 2010, PFCA time series can vary in the same species from different Arctic regions. Polar bears is a good example, as well as seals. But generally, long-term trends were declining up until 2010. 2010 to 2017 until present, there is more data about trends, particularly from the Canadian Arctic, East Greenland polar bears, Arctic foxes from Svalbard. And those increasing trends are connected with continued emissions of PFCA precursors and perhaps inflows from the Arctic Ocean, especially the North Atlantic. And then the fact that there are various confounding factors that are shifting those trends or levels in exposed Arctic wildlife. Finally, again, some more thinking to do here. This is an excellent review that came out by De Silva et al., an ET&C, that talked about PFAS exposure pathways for humans and wildlife. And what we can see here is a reiteration that what was found in that review was that wildlife exposure assessments are limited to targeted PFAS up to about 30 compounds, again, mostly perfluorinated acids. Forever chemicals, PFAS is dominant. Replacements, as I mentioned, there's still little data in wildlife and mostly non-detectable. So obviously something being told to us here, but also that we need more data. Also, we also know that PFAS, as they conclude, production is shifting rapidly. Targeted PFAS methods are lagging behind. Total fluorine, complemented by suspect screening using high-resolution mass spectrometry, are essential tools for PFAS exposure assessment. Also considering that the changing complex mixture nature of PFAS and exposed wildlife, and you can see to the far right, they have a nice plot there that shows basically how much of the total fluorine in a sample is not accounted for by targeted analysis. And finally, they talk about bioaccumulation processes differing substantially between PFAS and neutral lipophilic organic compounds, basically more PFAS generally being more protein associated, that we have to understand better those drivers that lead to accumulation and exposure levels in terms of, for example, protein exposure and processes. And with that, I would like to thank you. It's a lot to pack in 30 minutes, but I'd be happy to entertain any questions you have as part of my presentation or as part of the group discussion. Thank you very much.
We have Rob with us. Rob,
hello.
yes. Hi, thank you for your presentation. And Pierre has been pasting the questions in the WOVA app, but perhaps we'll take any live questions right now. Anna?
Yeah. Do we have any questions from here in Providence that we can pose to Rob?
Well, I think you asked one.
I did. I wasn't sure if anything was going to present itself here, but yes, I asked a question during your talk. Just a fantastic talk, Rob. So
Thank
you
you.
spent some time discussing the different monitoring programs that are in place, and there's just such a wealth of information that's come out of those programs. Can you maybe comment on how you see those programs could flex or change in the future as we kind of keep addressing some of the emerging PFAS questions you highlighted at the end there?
Yeah, certainly. Well, I mean, everybody knows with monitoring, there's this a bit of a conundrum between what you should monitor and what you can monitor, whether it be methods or whether it be simply the growing list of chemicals that you need to monitor. And you know, for a lot of these replacements, PFAS replacements that I mentioned, at least in Arctic biota, they tend to be very low level or non-detectable. So that, like a monitoring program in Canada, like NCP, has to make a decision about, well, should we be monitoring something that's really low level or non-detectable, or should we focus on chemicals, and many of which are PFAS, that are showing up at considerable concentrations, and retrospective trends are indicating perhaps increases. So I think that's sort of the, well, It is a challenge for monitoring programs, not just in the Arctic but around the world, as to and in the context of this increasing number of PFAS that may or may not be of environmental concern. I don't know if that answers your question or not.
Thank you so much.
I'm going to go into this next question, Rob, and I think it's a good one. So you presented a lot of data on PFAS in polar bear liver. And of course, the motivation for the NCP program is the exposure of indigenous populations in the north to contaminants through a traditional diet. And as far as I know, the Inuit do not consume polar bear liver. So this kind of ties into Reiner's question about, is there any inkling on the importance of neutral unknown PFAS for bioaccumulation and indication of more PFAS in fatty tissue such as blubber, which is an important component of diets?
Mm-hmm. Yeah, that's the basis of the human top consumer country food predator relationship. For polar bear, and because we had tissue samples for East Greenland polar bears, a couple of years ago we did do a tissue distribution study, and levels, for example, of again carboxylic and sulfonic acids are really, really low in polar bear fat. It's really the liver and blood, and to some extent brain tissue. So yeah, it's and I believe the same is true as, for example, ring seals or other seal species where there's been some blubber liver comparison. So obviously in terms of human consumption, there's a huge bias in terms of what tissues and what concentrations are of bioaccumulative PFAS, but in fatty tissue, generally much lower levels.
I wonder too, Rob, if some of that has to do with our analytical methods, right? Like all of the methods, we have this issue with water analysis too. The methods have been developed to target a very specific suite of PFAS.
Indeed, you're right. That of course is the challenge too. Is that we really need to expand more into what are more probabilistic bioaccumulative PFAS, like you say, the neutral compounds, because obviously we have a blind image of that when it comes to a lot of wildlife.
Absolutely. So as we're kind of wrapping up a question session here, do we have other in-person questions here in Providence?
Oh, okay.
I see Angie.
I was going to go. So I just wanted to, as an observation as a biomedical scientist, I think what's interesting is it doesn't seem like the industry is learning about the chemical structures and where the newer novel PFAS end up. And what I mean by that is that they're often acidic in nature. And so if they're acidic, their chances at a gut pH, they're not really going to be absorbed through permeability. They're going to be absorbed through transporters and other mechanisms. And so it's not kind of surprising that again and again, I know there's other mechanisms like FABPs and albumin that contribute to this, but in general, we're seeing the same patterns because in some ways they're all generating acids. Now, I know the neutral compounds are different. In our hands, when we measure them in cell-based permeability assays that are used in pharma, we do see that they're much more permeable than PFOS or PFOA. And so that will contribute to kind of, Ian's, I guess, Reiner's comment too. But one that sticks out at me that I didn't see was PFBS. And I wonder if it's because it kind of has some of the qualities that would make it not stick around and not stay in things. So it doesn't really bind to proteins well, whether it's albumin or other tissues. And even though it leverages transporters, it also leverages the same transporters in the kidneys to get out. So it gets in and it gets out, and it doesn't stick around. So I just wonder in some ways if maybe the engineering needs to kind of go in that way versus some of these molecules that are no matter what going to kind of do the same thing. They're high molecular weight, they're acidic, and they're just going to use transporters and stick around and deposit in tissues like the liver and the kidney where those transporter systems exist, but there's not really a good mechanism to get them out of the tissue.
Yeah, that's certainly what you say is very important. It's a lot to unpackage, but well, I mean, let's face it. The industry shift has been large. A big shift to shorter chain, and obviously with shorter chain, you have something that is, if it's longer chain, it's more lipophilic, like fatty acid, and then as you decrease that chain length, you have something that's more, more polar or more ionic, and of course that would change the dynamics as you point out. But yet, at least, again, if you go above fish, perhaps in terms of biota, I mean, people are looking for those shorter chain, like perfluorobutane sulfonic acid or perfluorobutane carboxylic acid, and simply not detecting it. I mean, maybe what's trading off there is again, there's a relatively low, let's say, burden, in which there can be exposure and accumulation, and at the same time, like you say, those shorter chain acids, for example, they're less. They have more specific mechanisms by which they would be retained in the body, and perhaps differential in terms of where they'd be deposited and also depurated. So, yeah, that's. I know what you're saying about lessons learned by industry, and we all know that it's just easier to make the same thing and just kind of tweak chain lengths, make them linear, make them branched, what have you, and get the same properties. And of course, that's also reflected in some of these important replacements we're seeing, like Pfas, just changing the general structure. So I think your question also applies to new replacements in general. I mean, there seems to be this minor tweaking conveyor belt concept going on.
Thanks so much, Rob. We have one more in-person question here in Providence.
Perfect. And my question is along similar lines. I found it intriguing, the new replacement compounds that you're not finding in the wildlife. I understand that Gen X, that's one of them, is eliminated in the body pretty quickly. I was wondering if Adana is the same way. Is it eliminated in the body, and that's why you're not finding it in wildlife?
Well, it would just be my opinion, but I think that's part of the issue. Part of the mode is it's not retained as effectively. Those really short chain fluorinated substances and acids are not being retained like the longer chain.
If I can also add to that, so definitely one hypothesis is that it's just not as bioaccumulative, but we also do a lot of environmental sampling in the Arctic, so water, ice, air, and we're not finding it in those matrices either, which kind of suggests that they're just not, maybe they might have a lower long-range transport potential than some of the other substances we're monitoring. Mm. I think we have time for another question. I'd like to read out Melissa Woodward's question. I really like this. So the question, Rob, is, are there any studies that look into how animal migration may impact the movement of PFAS into or out of the Arctic?
Yeah, well, for polar bears, of course, if I pick on polar bears, because I like you say, I'm the polar bear guy, when we look at those animals, we do look at very specific subpopulations which contain that subpopulation's migratory behavior. So like West Hudson Bay. I mean, the bears are moving around there, but it's still, in a bigger context, it's still a contained bubble of migration, although you get some individuals that go off in all directions. But that is also the challenge, is to try to figure out, in terms of field sampling of biota, or particularly wildlife, to have a good understanding of how they are moving around and at the same time are actually changing their dietary habits, right? Whether it is intentional or not, of course, that's where the shifting profile in the food web below like an apex predator is also shifting. So you have that confounding factor where you have that migratory element perhaps, but then you have the shifting food web because of movement of creatures lower on the food web. And again, that's why we pick on Hudson Bay when it comes to the APEC polar bear for that reason, and try to unpackage that. But yeah, I think when you talk about migratory behavior, you're going to have to also tie that in with dietary shifts, right? Because there's obviously some change in whether it be intentional or opportunistic, right? So I don't know if that answers your question, but that's what comes to mind.
That certainly is a great question. There's still so much to do. With that, do we have any other in-person questions here in Providence? I'm not seeing any takers. Amelia, would you like to keep doing any of the HUVA questions, or shall we move on?
I think we have time for one last question before we hear from Jinsia. And so this is a question brought to you by Mark Striner, and it's a bit of a doozy. So the question is, while global monitoring of many PFAS is important for widely dispersed PFAS, it seems to be equally important to monitor for select less globally dispersed PFAS in select locations. Global dispersion is not always that important for communities exposed to select PFAS at discrete locations. How do you respond to this?
Yeah, that's a lot to unpackage, but I think if you kind of pull it into a monitoring context, and like any sampling we do in the environment, it's a snapshot, right? We're also trying to layer onto that what is the production, what is the number of sources, the tonnage that gets into the environment, and just because you don't measure something at any point in time in a particular organism doesn't mean you won't detect it later. So again, I think that's why you see monitoring programs for kind of slotting monitoring for such compounds that are not detectable, low, but maybe heavily dispersed. There's a revisitation element, say every 2 or 3 or 4 years, just to see what's happening, especially if it's known that the source production is increasing and it's becoming a more strategic replacement, for example, in the context of PFAS. So yeah, and I just keep thinking of the trade-off here, because monitoring is such an expensive endeavor. You have to be strategic both in what you're monitoring and also where you can actually see define an exposure, whatever detection limits you might have, to be able to say something about changes in time and therefore make some judgment about whether it could become a problem in future. So in that, I kind of think of global dispersion and PFAS in that context.
Okay, great. Thank you so much, Rob. Please stick around for the panel discussion that will be at the end of this session. The speaker is Professor Christopher Solis, who is the Director of Environmental Science and Studies Program at Towson University in Maryland. Christopher will be speaking about ecotoxicology.
Good morning, everyone. Hope you're all well. My name is Chris Solis, and I'm here to talk to you today about some perspectives on ecotoxicology of PFAS. And I would like to thank the organizers of FLUOROS for inviting me to give the presentation. So overall, I want to talk about ecotoxicology, but basically in the context of ecological risk assessments. It kind of frames my perspective with regard to ecotoxicology. And then we'll talk more specifically about ecotox of PFAS. And one thing I think that's worth emphasizing and thinking about is that I think these chemicals are fairly complex, or at least that's been my experience in terms of and being able to work with them. And so I think that's a theme that's kind of emphasized or at least touched upon throughout. And I'll mention that my experience with PFAS started doing a kind of site characterization, if you will, of a Air Force base where there was former fire training areas, and we'll talk a little bit about that. And so there was contamination of the local water body, which was a bayou. And that was sort of my introduction. And then since then, I've done quite a bit of work related to ecological risk assessment and ecotox of PFAS. One point worth mentioning is there's this paper that was published by Ankley et al. This year basically focused on ecological risk of per and polyfluoroalkyl substances, published in Environmental Toxicology and Chemistry, and it was, in essence, the culmination of a workshop on this subject. So there's lots of information there. I'd encourage you to check it out. So ecological risk assessment, as a lot of you probably are aware, is basically the way that science is used to help make decisions about how to manage the environment, mostly with regard to chemicals. So the ultimate goal is reasonable environmental protection. And ecotox plays a critical role because risk is, in essence, a function of toxicity and exposure. And so the ecotox part is this toxicity element. And then for the most part, the way that most risk assessments are conducted, there's an emphasis on what are called apical endpoints, and those are either survival, growth, and reproduction, or traits or characteristics that relate directly to those. And that's because these endpoints are more easily translated to effects at the population level, and it's often the population level that's a focus of management and regulation and concern. So PFAS, as a lot of us know, in the environment is a complex story from the outset. And that's because, in fact, there are many, right? And this great paper by Wang et al. In 2017 kind of highlights that, and that there are many thousands that were in or could have been used, and then many that still are. When we look at the story of ecotox, with regard to PFAS, two kind of emerge as being sort of most interest, and that's PFAS and PFOA that a lot of you, I'm sure, are familiar with. And there are reasons, in part, I think that a lot of concern has focused on these chemicals because they've been measured in a wide variety of media and tissues of organisms. But just to kind of give you a sense, right, I did a quick Google Scholar search using, say, PFAS and ecotoxicology or PFOA and ecotoxicology, and just reported the number of references that popped up. So just gives you a perspective, right? Clearly, PFAS, PFOA have been well studied, and PFAS even more so than PFOA. And then if we look at 6 chemical sulfonates and carboxylates, we see that substantially less, but some, right? And so, it's very much the case where there have been a few PFAS that I think have been studied really well, and a few that have been studied, maybe more that have been studied somewhat, and then more that we really just don't know about. So when we think about ecotox, clearly we have to consider, well, which PFAS are we interested in? And my research, I mentioned, started with looking at Barksdale Air Force Base, and a lot of Air Force bases and airports had used what was called aqueous film-forming foam to extinguish fires and for fire training exercises. And these had relatively high concentrations of PFAS. And then when we look into the environment, right, contamination that has occurred as a result of those activities, we often see it's dominated by a few PFAS. So we did this paper, or published this paper in 2021, where we had a database of surface waters at many Air Force installations, U.S. Air Force installations, with measured PFAS concentrations. And what we found was there was really dominance by PFAS and then followed by PFHXS. So that's the 6-carbon sulfonate. And the other interesting thing, we sort of looked to see if there was a sort of identifiable common mixture. And it turns out that there wasn't, except that if we considered a 4-chemical mixture, that often did a really good job of sort of characterizing 80% or more of the sum PFAS. So most of the PFAS in a mixture of PFAS, at least in these sites, could be described by like a 4-chemical mixture. But in that, again, PFAS and PFHXS were very dominant. So the point of this exercise was, in fact, to kind of help inform ecotoxicology experiments moving forward, particularly those focused on risk assessments of these sites, which are somewhat, contaminated. So there was another nice paper published by Ahrens and Buncha, I apologize if I mispronounced that, in 2014. And really this kind of also kind of summarizes an important perspective, which is that across many wildlife taxa, there are clear demonstrations of PFAS and PFOA in their tissues, clear, evidence that they have been exposed and accumulated these chemicals to some degree, right? So we have a lot of information that has pointed to a few chemicals, PFAS in particular, being relevant, both in the environment, and in wildlife. But that it's important not to exclude other PFAS because we're getting more and more data and better analytical techniques. But the other element that we want to think about when we think about ecotoxicology is the nature and duration of the exposure, right? So in ecotox, studies fall kind of more or less into two categories: acute, which are fairly short-term, high concentration exposures, and chronic, which are longer in duration and tend to be lower concentrations. So there are, for all intents and purposes, PFAS have not really been shown to be that acutely toxic. And moreover, I don't think that there are a lot of instances where there are really high concentrations in the environment. So this is an example from Moody et al. Where there was an accidental release of foams at an airport, and certainly there was a spike in concentration soon after the release, but then through time, right, that decreases. And in fact, when we look at longitudinal data, so data through time for PFAS in the environment, more commonly we see sort of lower concentrations that are, somewhat constant, if not decreasing a little bit through time. And so this Munoz et al. Looked at an estuary in France. And this is reminiscent of data that we've collected, both at Barksdale Air Force Base and other Air Force bases, looking at surface water concentrations. So relatively constant with a little bit of fluctuation through time. So when we think about ecotoxicology, then those types of studies, sort of longer duration, lower concentration, are probably most relevant. Okay, so I'm going to use PFAS, even though I know a lot of folks are interested in other PFAS, but the PFAS ecotox story at this point is really has focused on PFAS, and I still think it's very relevant. It is arguably the most studied PFAS, and in general, it's the most toxic. When compared to the toxicity of other PFAS, it often emerges as being toxic. So there is tox data for a number of species from different taxa, but I'm going to use invertebrates and avian species to kind of illustrate some points. So McDonald et al. In 2004 published a very nice paper on chironomids, which were, and maybe are still, among the most sensitive invertebrates that have been tested. And they did 20-day exposures, which are, is more or less the duration of the aquatic life stage of the species. So it, starts as an egg, goes through a larval stage, and then emerges as an adult. And that 20-day exposure captures most of that process. And when we looked at, if you look at survival over 20 days, you see a very nice dose response, right? As concentrations of PFAS increase, survival decreases. And similarly for size or weight. But what's interesting is at the lowest PFAS concentration, now we see a significant effect, but that effect kind of goes away, if you will, at intermediate concentrations. And this is not an uncommon kind of response where it's a non-monotonic exposure response. And this is one of those factors, I think, that makes the chemicals challenging, is that it's not that uncommon to see this sort of non-monotonic response, especially at low concentrations and longer duration exposures. So they also included emergence as an endpoint, and this is the control up here, and then all the rest of these are the PFAS or PFAS exposure concentrations, and we see a pretty strong departure from control. So pointing to kind of another endpoint, and this is a even longer, ended up being almost 40 days. So McCarthy et al., and I was part of this paper as full disclosure, in 2021 published kind of a repeat, if you will, of the McDonald et al. Work using the same species, but also looking at 5 other PFAS. And this was a fairly convincing data showing that indeed PFAS was substantially more toxic than all the rest of these other PFAS, right? The other point is, and I should have mentioned it previously, but McDonald saw effects on chironomids at concentrations below 10 micrograms per liter. In fact, most of them, I think, were below 5 micrograms per liter. And in this paper, we saw similar effect levels, right? Concentrations in the neighborhood of what McDonald et al. Reported. And so this, from an ecotox perspective, is another point worth mentioning, is that the more data that we have, even if it's repeated for the same species, similar design, if it corroborates previous studies, then we have more confidence in those observed effects at those concentrations. So this was really helpful. Now, so another point worth mentioning, and why I brought, I'm using aquatic invertebrates kind of to illustrate another point, which is very long-term exposures. And so not easy to do multi-generation exposures with a lot of species, but not so difficult with aquatic invertebrates, or at least I should take that back. It can be very, time-consuming and requires a lot of very skilled laboratory technique and methods, but it can be done. And this was a great study, Marziali in 2019, 10 generations of exposure. So parents were exposed, the offspring reared in those the PFAS environment, and then the exposures were more or less continuous for PFBS, PFOA, and PFAS. Okay, so what the details are not important. These are different at this stage. Are different life history traits, like related to reproduction and growth primarily. And the point is that there's no real strong pattern through time or within the chemicals, right? There are occasional significant effects for different endpoints at different generations and for different chemicals. And one thing that is not evident is sort of an accumulation of effects through time, right? If parents and offspring are continually stressed, we might hypothesize that through time we'll see even more significant effects. But that's not really evident here. And in fact, when all these data were combined into a population model to estimate population growth rate, there was no signal of an effect from any of the PFAS. The other thing that's really interesting about these studies, we can ask, well, how does this change their tolerance, right? Multi-generation studies have the potential to have kind of an evolutionary effect where you're changing the tolerance of species, and so in this case, pre-exposure, so that 10 generation of exposures did not have, well, it had an impact on their tolerance to a more acute stress. They were actually more sensitive after being exposed for a pretty long duration. So super interesting results. And I think there was some follow-up work related to this, but really, really neat. And there's some other data for other invertebrates that, again, are kind of in the neighborhood of effects that I just talked about for chironomids, somewhere below 10 micrograms per liter for PFAS. And so this kind of gives us some confidence in terms of where we think effects may occur. So now I want to segue to birds, which is a really interesting story. And in part, I want to talk about birds, even though I'm not an avian toxicologist, although have participated in some studies. But avian species are common receptors for ecological risk assessment, and they're of high interest to society, right? There's a lot of field data for birds, so not just concentrations of PFAS in birds, but also were there effects in birds that were in the field. And so that's really helpful to us. There are also several laboratory toxicity studies and several ecological risk assessments with birds as receptors. So it's kind of a soup to nuts story with birds that can be very helpful. And I'm going to highlight several studies I think are really interesting and tell a really interesting story. So Christine Custer at USGS, we actually have a project that I hope we're going to be starting, but in 2012 and 2014, published some really neat papers showing these are wild tree swallows that hatching success decreased with increasing Pfas concentrations in eggs. And in fact, the point of departure was around 148, 150 nanograms per gram in the egg. 2 studies in a similar area, right, similar part of the United States, but 2 different studies showing effects at a fairly consistent concentration in eggs. And so to me, for a while, this was really compelling evidence that PFAS, in this case, could cause effects in wild species, right, that are being exposed naturally, if you will. So in 2019, she published a paper where they did the same sort of experimental design looking at tree swallows at Wurtsmith Air Force Base, in part because there was really kind of just PFAS contamination. So the other sites I should have mentioned were more industrial, urban, and that can be kind of a mixed bag. And even though they did some characterization of other contaminants, it's still a more complex, probably stressor environment than what Wurtsmith was, which was primarily PFAS, but also, as I understand it, pretty high habitat quality. And they saw PFAS concentrations in eggs much higher than previously, but really no effect on hatching success. And there are a number of hypotheses for why this might be, including, there are other contaminants in the industrialized area that may be responsible for the effects that can co-occur with PFAS, maybe habitat considerations, or who knows, an interaction of several factors. But it's a really, really interesting story. And again, points to kind of that head-scratching type complexity associated with understanding what's going on with these chemicals. And then here's kind of another example. So PFAS in lab toxicity using birds, and I'm going to focus on bobwhite quail. And here again, the story is mostly about PFAS. There's another study on mallard ducks. And I've been part of a research project that looked at PFAS other than PFAS in terms of toxicity to birds using a kind of abbreviated life cycle design or reproductive study where adult birds are exposed and then they're allowed to reproduce, and then effects are observed or investigated in both parents and offspring. And here again, right, we looked at all these different PFAS, and the only ones that were toxic were really PFAS, the combination of PFAS and PFHXS, and then kind of a new player, PFHXA. So Newstead et al., this is kind of where the story begins, published a series of papers. One was the toxicity data, and one was kind of a summary and synthesis of those data to be used for risk assessment, right? And for bobwhite quail, they saw effects at this, single-digit ish milligrams PFAS per kilogram body weight per day. So that's a common exposure metric, right? So they importantly did a dietary exposure. So more recently, Dennis et al., and this is the work that I'm a part of, did drinking water exposure. So a very different mechanism of exposure. It's still an ingestion exposure, but one is obviously in food and the other is in water. And here, hopefully you can see that, but in essence, same units, milligram per kilogram per day, same adult approximate daily intake, right? And concentrations were much, much lower, orders of magnitude lower in terms of effect levels. And one of the effects that kind of emerged as being important was this sort of pipping but not hatching. So hatchlings breaking the shell but then not being able to hatch completely. And so that's certainly a very interesting effect, and harkens back, if you will, to some of the work by Custer et al. Showing decreased hatching success. And in fact, the concentrations that occurred were similar in the eggs. So then Dennis et al., Nicole Dennis and Todd Anderson at Texas Tech explored PFHXA, for which there is not many data. And that the decision to do that was based on an earlier work that I mentioned, actually, where we characterized PFAS in surface water. And PFHXA emerges as being somewhat common and potentially important in some cases. And interestingly, what they found was that PFHXA, the effect levels, were lower than even PFAS. And pretty much all of the effects were focused on weight or chick growth. And in fact, this effect on size I've observed numerous times. We've conducted a series of studies on reptiles that we hope to get out soon. But there, the dominant effect seems to be on size. And when you look into the literature, when we looked at, we've also done some studies with fish, same thing. We see the kind of most significant effects on size or growth. And again, here, it's echoed as well that PFHXA, in this case, much more toxic than PFAS, and pretty much the main effects were related to growth of chicks. So, it's a really interesting story, and I think it's continuing to evolve. That is the ecotoxicity of PFAS to avian species. And I think there are multiple lines of evidence now that point to effects of PFAS on growth and or reproduction, but not without some uncertainty, right? And that, again, echoes that notion of complexity. And for the first time, actually, we tested a PFAS other than PFAS that appears to be more toxic. Now, we need more data, right? I mentioned that the value in repeating that Chironomids study bolsters our confidence in terms of potential for effects and our confidence in risk estimates. I think that this is true in everything that we do, but clearly if we identify a chemical as being very toxic or more toxic than PFAS, then we need some more data to kind of improve our confidence in that. But it also points to the importance, and this shouldn't be a shocker, but the data illustrate that the exposure route is likely very important, and then there could be a bioavailability issue where PFAS in water, and they're somewhat soluble, PFAS in water are much more available to the organism if they're taken up via water than compared to food. And we're following up with that research question in terms of how does the ingestion route, whether it's dietary or drinking water, how does that influence toxicity? It's really kind of interesting. The other sort of giant in the hills, if you will, is the idea of mixtures. And I didn't get into too much detail on that because, first of all, there's not a lot of data. There is a lot of data showing that PFAS occur commonly as mixtures in the environment. And I talked about that when I mentioned our work that was published in 2021. But I think that there's a well-recognized need for a better understanding of the ecotoxicity and the toxicology in humans of PFAS mixtures, right? To start, there's no real meaningful consensus, as far as I'm aware, on the mode of action, right, of PFAS. And so that makes kind of progress with regard to mixture toxicity a little bit more challenging. And then when we look at the available data, we actually see a range of different types of interactions, all the way from antagonism, which would be this case here, where the data, the purple line, are below this kind of envelope of additivity, which is between the green and the blue lines. A paper published a few years ago out of Purdue. This suggests antagonism, where there's less toxicity with two chemicals interacting. And this is for PFAS and PFOA. And if you look at other ones, most of them are additive, right? And this is for, I should have mentioned amphibians. I forget the species. But there's also, a series of other papers related to the mixtures of PFAS that have seen within the same paper everything from antagonism to synergism, where there's greater than additive toxicity when you combine two PFAS. In our own work, we looked at PFAS and PFHXS mixtures because, again, at a lot of Air Force bases, those two chemicals are particularly prominent. If we look at lizards, so we did this work in my lab, the combination of PFHXS is not toxic. We've never observed a toxic effect with PFHXS, lizards exposed to PFHXS. We've commonly observed toxicity with PFAS, and we feel very confident because we've seen it several times, effect levels at the same concentrations, same effect. But when we combine PFAS and PFHXS, right, PFHXS, there's no toxicity by itself, we see greater toxicity than PFAS alone, right, suggesting a synergism. And this is true when we looked at fish as well, same sort of design. So when we move to crickets, so we have a cricket model that we've been working on for Ecotox, and we were going to do this, we did the same design. I was very certain we would see the same thing, which is greater toxicity when we combine PFAS and PFHXS, which is this treatment. And in fact, though, we saw very strong, very clear antagonism, as if the PFAS didn't exist. I mentioned that in the previous slide with the study on amphibians. Again, we need to repeat this cricket study because it is a very surprising result. But these data, having looked at many cricket data, are solid. And so I'm inclined to believe them, but we need to look further and kind of better understand and repeat this study to get a better sense. But again, it points to the complexities associated with these chemicals. So when we think about future efforts, I still think that even though we can measure many PFAS and we should pay attention and conduct research more than just on PFAS and PFOA, I still think that PFAS is going to be an ongoing concern from a risk to wildlife perspective. I think PFAS mixtures and getting a better handle on that is definitely a high priority. We might want to, and folks are, and we should also look at some of the longer chain PFAS, particularly in remote areas. Like if you look at data for bird species in the Arctic, they often have high concentrations of longer chain PFAS in their tissues, and there may be some effects associated with that. Progress with regard to mode of action, I think, is probably going to take us to a point where we can better understand PFAS mixtures and the so-called new approach methods. And that's where basically using high throughput toxicity testing or suborganismal responses to kind of better understand and characterize toxic mode of action. And so I'll point you to a paper that was published in 2019 where they were prioritizing 75 PFAS, and this is an EPA researchers or in conjunction with EPA researchers. And in essence, these then would be the subject of high throughput toxicity testing, looking at a whole suite of relevant endpoints. And with that, I think I'll end. I want to acknowledge the funding that I've gotten through the years, but also especially the FLUOROS organizers for giving me this opportunity, and then a whole slew of collaborators that I continue to enjoy working with, and I'm hopeful that we can make some positive contributions. And with that, I think we'll segue to the question and answer. Thank you.
Thanks so much for that awesome talk, Chris. I'm sure that data really made quite an impact on the audience. Some very head-scratching aspects that you highlighted there. We have time for a few questions. One of the questions in the chat, I think it's from Anna, and I had the same question about the multi-generation. You presented the data on the Chironomids, 10 generations that showed increased sensitivity in that 10th generation to PFOS. So I'm curious, obviously it's hard to do those multi-generational studies in other sort of higher trophic level organisms, just based on the life cycle. But I'm curious about when you say things like that PFHXS is not toxic based on the studies, is it possible that if you continued that study for multiple generations that you would possibly see an effect in some of the further generations.
Can you hear me okay?
Yes?
Yeah. Yes. I certainly think that it's possible. Can't necessarily exclude it. So, and I'll just mention that even for, although we haven't in a range of taxa over what would be considered reasonable duration exposure length, again, we haven't really seen any effects in birds, fish, reptiles, chironomids. That said, invariably, if we increase the duration of exposure with PFAS, we start seeing, effects at lower concentrations. So I can't exclude it with regard to PFH excess. Yeah. But, the multi-generation study suggests that at least under those exposure conditions, there wasn't a real strong signal of, kind of an increasing effect with time. That doesn't mean, right, I've said from maybe the first slide, it's complicated. That doesn't mean if you did the study in another species or even the same species, I wouldn't guarantee you would see the exact same result. Yeah, hopefully that helps.
Anna, would you like to field some questions from the live audience?
Absolutely. Do we have a question in the back? Oh, that is not a question. I have a question, and this is, I don't know that much about invertebrates. So you mentioned you focused on the chironomid data quite a bit. You mentioned those are some of the most sensitive to PFAS impacts. Can you comment on why compared to other invertebrates in particular?
Well, I have some thoughts. So in that McDonald paper, they actually, if you read it, they report that exposed animals were kind of behaving sort of oddly, writhing on the surface, and their oxygen carrier is hemoglobin. And so they hypothesized that there was some interaction or effect related to that. And so that is an interesting hypothesis. My evolving hypothesis, I mentioned that growth is something that like an effect on growth is something that we see, I would say, commonly. And that suggests a metabolic kind of effect or mechanism that results then in a decrement in growth, right? Somehow the chemical is monkeying with their energy budget in some way, shape, or form. That kind of relates back to, hemoglobin as an oxygen transporter because it's clearly important for metabolism. So that's all circumstantial. And my kind of hypothesis looking at just data across a range of species and a common endpoint and why we would see effects like that. So, I think the answer to the question is, well, there's something specific about their physiology or life history that, kind of makes them more sensitive. And we see this, this is not an uncommon thing in Maryland streams, probably in streams in Canada too, right? You look at certain taxa, mayflies, caddisflies, stoneflies, in particular, as being indicators. If they're there, then that generally means your streams are higher quality. And so that's like a metric we use. So there are differences among the taxa, even within aquatic invertebrates that make them more sensitive or not. And with chironomids, that's my kind of off-the-cuff hypothesis.
A few questions. Actually, there's a lot of questions in the app, and one of them sort of relates to something you mentioned in your response there. So as we look at other PFAS beyond PFOS and some of the likely candidates, beyond some of these growth and metabolic impacts, what other kind of endpoints should we be keeping an eye out for in your estimation?
So I think that there's a pretty strong consensus that there's immunotoxicity. And I think there's, right, the exact mechanism of it may be unknown. So I think that is one to me that is not immediately survival, growth, reproduction, right? That would go into, say, your assessment of risk, but it relates pretty strongly. If, an organism were to interact with a pathogen, which they almost invariably do. So there's another one that I think is close to apical, if you will. But I think that I really have hope for some of these high throughput toxicity testing approaches that will help point the way to kind of mode of action, but then also what endpoints at the organism level, how that all kind of works together. So maybe folks are aware of the adverse outcome pathway kind of framework. If we understand what's happening all along the way from the point of a chemical kind of entering an organism and the molecules within the organism that it interacts with and how it affects those processes that are, occurring within the organism that you can't necessarily see, but then more importantly, how those then translate to effects on survival, growth, and reproduction. And that's really what we're after. And so far, what I gave you was really just that, well, let's look at survival, growth, and reproduction, because if we needed to make a decision and do a risk assessment right now, those are the data that we would generally use. But as we move forward, we're going to have to start addressing that question using some of the fantastic tools that we have available to us.
So we have just a few minutes before Pierre's presentation, but I see that there are so many questions in the Whova chat. So what I would recommend is we take one more question, and then we can maybe revisit some of these other questions during the panel discussion. And Pierre, you can always respond to these questions in the Whova chat directly also. So Anna, one more question, please.
We do have one more in-person question here from Reiner.
Thanks a lot, Chris. Very fascinating and not very clear where this all ends. But my question was the drinking water part.
Yeah.
And I'm not an eco person, so excuse my ignorance here. Is that relevant for any species? I think most birds probably take it from food. Most insects might actually inhale things more than... So how does this fit in the real picture?
So that's a great question, because that other aspect of estimating risk is clearly exposure. And if you have to do a good job with both of those to have any confidence in your risk estimates. So it's a great question. I mean, certainly it's possible that wildlife do drink, and birds do drink water, but they also do get water from from food items. So in fact, that is something that we need to work on. And as we start crafting the final report and probably papers, I hope, related to how you use all this information to assess risk, that's one of the elements that we really need to think about critically. Because, via that specific ingestion route, it's much more toxic. But how relevant is that? And so that, in this case, requires kind of an exploration of data and assessment approaches to sort of figure out, is that even, how relevant is that? And that comes into, we can estimate that based on what we know about avian behavior and habitat. So it's a very good question.
Okay, thanks very much. Stick around for the panel, please. And the next talk, which is the last talk of this session, will be by Pierre Labadie, who is a research scientist at CNRS in Talence, in the Bordeaux region of France. Pierre will be speaking about bioaccumulation, field-based and laboratory investigations of bioaccumulation of PFAS. Thank you, Pierre.
Good afternoon, everyone. My name is Pierre Labadie. I'm an environmental chemist at CNRS, based at the University of Bordeaux in France. I've been working on the fate of PFAS for over 10 years, and I'd like to thank the organizers of the FLUOROS conference for inviting me to give this talk about bioaccumulation of PFAS in ecological systems, with special attention paid to aquatic biota. This is the last presentation of the session, and as I said, I've chosen to focus on aquatic ecosystems. I will address the following issues: PFAS occurrence in aquatic environments, exposure routes and processes, accumulation metrics, and modeling approaches. The accumulation of Pfas is a fundamental process that controls their internal dose in biota. This is an issue of major interest addressed by several hundreds of papers. The figure below shows the trends observed in recent years, and such studies provided a worldwide coverage for wildlife. They included a large diversity of habitats, like freshwater, estuarine, or marine ecosystems located in tropical, temperate, or even polar areas. They also covered a wide range of aquatic biota, like invertebrates, fish, reptiles, birds, and mammals. Here I just give an example of Pfas occurrence across the globe. This figure was released in 2011. It shows the levels of PFOS in the liver of marine mammals worldwide, even in remote polar regions. PFOS was reported at very high concentrations that result from bioaccumulation or even biomagnification, two processes that have been extensively investigated of PFAS, as we will see thereafter. Diverse PFAS are used in a variety of commercial products and industrial applications. So far, the vast majority of studies on aquatic biota have focused on well-known PFAS, such as perfluoroalkyl acids, PFAS. Relevant legacy PFAS identified in biota include 2-sulfonates, PFHXS, and PFOS, often the most abundant PFAS in aquatic biota, but also C18 to C14 carboxylates, which are also very frequently detected and can be as abundant as PFOS in some cases. Varying ratios are observed depending, for instance, on the considered taxa or on the contamination pattern of the sampling site. More uncommon PFAS have also been detected in aquatic biota, such as short-chain PF carboxylates. Relevant compounds also include more reactive neutral PFAS, such as Phos or A2-FTOH, which may be transformed into PFAS. The global environmental contamination and the proven or potential toxicity of PFAS has led to regulation of some of them, mainly PFOS, PFOA, and long-chain carboxylates. As a consequence, there's been major changes in the industry, shifting towards replacement products such as short-chain PFAS, fluorinated phosphatesters or ethers, as well as perfluorinated cycloalkanes. Cousins and colleagues have reviewed this issue, and they suggested a list of potentially bioaccumulative PFAS, as shown here. A common feature of those compounds is that they contain at least 6 perfluorinated carbons. In addition, a number of emerging PFAS are already suggested to be bioaccumulative according to lab-based bioaccumulation experiments, like phosphonic and phosphinic acids that are slowly eliminated from model species like rainbow trout, similarly to long-chain carboxylates. There are also field-based evidence for the widespread occurrence of emerging PFAS in aquatic biota. For example, perfluoroalkyl chlorinated ether acids, which are components of the FT53B formulation, were detected in biota from the Chinese Bohai Sea, albeit at rather low levels compared to those of legacy carboxylates. Another example is given on this slide, which shows the occurrence of a cyclic perfluoroalkyl sulfonic acid, PFEX, in fish collected from a pond near a major airport in China at levels similar to those reported for long-chain carboxylates. Data on those emerging chemicals still remain scarce. To get a wider picture of PFAS accumulation in biota, complementary approaches are needed. A major challenge in PFAS exposure assessment is actually to establish the total burden of PFAS resulting from multiple exposure pathways. As we will see later on, approaches have been recently implemented to address this issue. They rely on targeted quantitative analysis, restricted to a predefined set of analytes, on HRMS-based suspect or non-target screening. See Yana Yu's talk for this issue. And also rely on emerging approaches such as the determination of extractable organic fluorine, EOF, or the total oxidizable precursor assay, the TOP assay. Non-PFAS are typically determined using target LC-MS analysis on the same sample extracts used for EOF determination, which are analyzed using combustion ion chromatography. The fraction of unidentified organofluorines in a sample is calculated by subtracting the concentration of target PFAS from the total organofluorine content in the same extract. Here I show you the results obtained from marine mammals sample near the US East Coast. In these samples, targeted PFAS accounted for only 30 to 75% of the EOF, indicating the massive presence of unattributed precursors. The TOP assay relies on the oxidation of reactive precursors into carboxylates that can be analyzed by LC-MS. Here are the results obtained with this approach on samples collected in an urban river near Paris in France. Black bars indicate that in this ecosystem, between 25 and 80% of PFAS samples in samples collected at the base of the trophic web were unidentified. As we have seen, PFAS are ubiquitous in biota. This is due to exposure through multiple pathways and accumulation results from several processes. This figure summarizes the major routes of uptake and elimination in fish: gill or dietary uptake, gill elimination, or fecal digestion, biotransformation. Growth dilution may be a confounding factor depending on the considered timescale and species. Compared to neutral hydrophobic POPs such as PCBs, the processes governing the uptake of PFAS into organisms and partitioning across tissues remain less understood, even for those PFAS that are more commonly investigated. In addition, the tissue distribution of PFAS is very different from that observed for other organohalogens. In fish, for instance, PFAS accumulate primarily in the blood or liver rather than in adipose tissue. There are currently two prevailing theories to explain PFAS bioaccumulation: interaction with phospholipids and interaction with proteins. PFAS have poor affinity for neutral lipids, but it has been shown that they have moderate affinity for phospholipids. Phospholipid bilayers through hydrophobic van der Waals interactions, hydrogen bonding, and electrostatic interactions. The first theory is therefore that partitioning to phospholipid bilayers has major influence on the whole body bioaccumulation potential of PFAS in aquatic organisms. Such interactions have been studied by in vitro and in silico methods. Results obtained by in vitro experiments revealed that both acidic and neutral PFAS could penetrate into phospholipid bilayers and occupy a substantial volume, as shown here for FOSA. The comparison between substances with the same functional groups but different chain length show that the penetration of PFAS into the bilayer increases with the PFAS chain length. The effects show to decrease with the functional groups as indicated here. However, phospholipid partitioning alone cannot really explain the trend of PFAS bioaccumulation for a wide area of PFAS, especially short-chain PFAS. Thus, another theory suggests that non-covalent interactions with various proteins like serum albumin, fatty acid binding proteins, and organic anion transporters are the most important mechanisms that govern the uptake, distribution, and elimination of PFAS. Protein binding is also driven by a combination of hydrophobic and polar interactions, as evidenced, for instance, in silico by molecular docking. Half-lives may be used as a surrogate for accumulation potential in different types of biological models. Here are shown heat maps illustrating the correlation between estimated protein and membrane binding affinity and measured half-lives in rainbow trout, pigs, humans, and rats, considering from left to right mixed linear PFAS, P4A isomers, and Phos isomers. No variable emerged as a single driver for explaining observed half-lives, and correlation suggested that both membrane partitioning and protein interactions were important. So, PFAS display complex toxicokinetics. Key features of these compounds include the tissue distribution, the relationship between bioaccumulation and perfluorinated carbon chain length, and their widely variable species and sometimes gender-specific half-lives. To mechanistically describe the bioaccumulation of PFAS, one should therefore consider both a phospholipid component to describe the distribution into tissues where little or no specific binding occurs, as well as a protein component in order to describe the accumulation in the blood compartment and elimination or reabsorption as mediated by transporter proteins. The accumulation potential of organohalogens is commonly reported using several metrics. Bioconcentration factor reflects diet uptake, the direct uptake of a chemical by an organism from water or air. The biomagnification represents the concentration of an organism relative to its diet. The bioaccumulation factor is related to the combined effects of all uptake pathways, and the trophic magnification factor is an indicator of biomagnification within a food web, as it can be seen as a diet weight average BMF. Finally, the BSIF is also used to describe and predict transfer from sediment to benthic biota, but these metrics won't be treated here due to lack of time. These metrics are useful to estimate the accumulation or magnification potential, to compare them across taxa and ecosystems, and also to compare them with regulatory thresholds. BCFs are measured in lab experiments and can be defined as the ratio of concentration in biota to concentrations in water, or as the ratio of first-order uptake and elimination rate constants. BAFs are typically field-based measurements. BAF is also defined as the ratio of concentration in biota to concentration in water, but uptake can result from respiratory and dietary exposure. These metrics are calculated on a wet weight basis, and Pfas, which BCF or BAF exceed a certain threshold, for instance 5,000, are considered as very bioaccumulative under REACH, and they fulfill the B criterion of the Stockholm Convention on POPs. As you can see in this table proposed by Burkhardt earlier this year, BCFs and BAFs are most often determined for fish, bivalves, and crustaceans. There's a relatively lack of data for species at the base of trophic webs, like for instance phyto or zooplankton, and most studies focused on freshwater biota. Other ecosystems, however, were also considered, but to a lower extent. BCFs and BAFs are usually derived from whole body burden, but levels in organs such as liver or fillets in fish, or even plasma, are also used in the literature. This results in whole body BAFs being slightly larger than those based on muscle, but lower than those based on plasma or liver. This figure, taken from a recent meta-analysis of the literature, illustrates this trend, with lower BAF determined on a field basis. See, for instance, Phos of PFA. Note that a wide range of BAF values was reported across species. In lab experiment, the BCF of some Pfas, such as Phos of PFA, seems to be inversely related to concentrations, but we get a mixed message from field data due to a fair amount of scatter. The potential mechanisms of this concentration dependency remain to be elucidated. They could be linked to active transport pathways. Though high variability is observed for some Pfas more rarely targeted in biota, relatively high consistency is observed across taxa and studies for the most frequently reported PFAs. BAFs, indicated here by yellow bars, are usually similar or higher than BCF, which is expected since BAF reflect multiple exposure routes. As it stands, there's a large amount of data for carboxylates and sulfonates, while there is a data gap for other Pfas classes like phosphate diesters or fluorotelomers. And the general trends for carboxylates and sulfonates are as follows. Highest BCFs are reported for long-chain carboxylates with 12 to 14 carbon atoms. For a same functional group, BCFs and BAFs increase with increasing fluoroalkyl chain length, and for similar chain length, sulfonates display higher BAF than carboxylates. In comparison with freshwater species, data are limited for brackish and marine species. While BCFs are higher in marine plankton, no clear trend is observed for fish as shown here. It's been hypothesized that bioconcentration or accumulation could be lower in marine waters due, for instance, to increased ion pairing with salinity or alteration or respiration rates. Pfas are present in the environment as mixtures, which has two main implications. The first one is competitive bioaccumulation. A few studies have investigated this issue, and I provide here an example taken from a study performed on fish under controlled conditions. The results show a decrease of steady-state BCF for short-chain carboxylates in different tissues, especially liver and blood, in the presence of long-chain carboxylates compared to that in the absence of long-chain carboxylates. The second implication of having Pfas mixtures in aquatic environments is that metabolic transformation can lead to the formation of PFAS if more reactive precursors are present and taken up by biota. Those compounds may be known measured, known unmeasured, or even unattributed precursors. This general issue was covered in Jean New's talk, and I just wanted to emphasize here the fact that there were a few reports for aquatic biota, especially fish. Here I give you a first example, showing the biotransformation pathway of disampap, a phosphate diester, which leads to the in vivo formation of Phos in European perch as a minor product. Another illustration is the in vivo biotransformation of F2-fluorotelomyl acrylate in rainbow trout, which yields the formation of PFOA as a minor product. As I said, PFA precursors may be found in aquatic ecosystems. For instance, in the North Atlantic, Spahn and colleagues recently reported on the occurrence of several classes of precursors using targeted and suspect screening methods. Such compounds included fluorothermal carboxylic or sulfonic acids, as well as sulfonamides. In a completely different ecosystem, an urban river in Paris, France, precursors were also ubiquitous. The figure on the left presents the levels of potential PFAS precursors, such as FTSA or DIPAPs. Such levels were not negligible compared to those of PFAS, especially in the case of 6,2-FTSA. In addition, as you can see here, the ratio of PFAS precursors to PFCA significantly decreased from the base of the trophic web to fish, with invertebrates displaying intermediate values. This strongly points to the strong biotransformation of precursors, with likely higher transformation rates at higher trophic levels. The scatter observed here for fish probably reflects the species-specific metabolic capacities, but it may also be related to species-specific toxicokinetics. In this freshwater ecosystem, the application of the top assay revealed the presence of very large proportion of unidentified precursors at the base of the trophic web, such as biofilm or sediment, accounting for up to 80% of total PFAS concentration. The proportion was lower in invertebrates, up to 50%, and in fish, up to 25%. And altogether, these results further suggest the occurrence of precursor biotransformation. Compared to BAFs, BMFs and TMFs are less often determined. However, in ecosystems, food web structures control the pathways and flow of energy to apex organisms, as well as the dietary transfer of chemicals. Understanding this phenomenon, known as biomagnification, is therefore crucial to further understand PFAS transfers in ecosystems. The biomagnification factor is defined as the concentration in a predator relative to its diet, may be adjusted to trophic level differences or to the relative abundance of different prey in the diet. TMFs are usually derived from regression analysis of concentration against trophic level, and their estimation allows comparing biomagnification across different types of ecosystems. They provide the most conclusive evidence of the ability of PFAS to biomagnify in food webs. Since lipid correction is irrelevant for PFAS, BMF and TMFs are usually expressed on a weight-weight basis. BMF or TMF higher than 1 suggests biomagnification, and this matrix should be used as lines of evidence for biomagnification assessment. BMFs are usually calculated based on field data, although they may also be determined in the lab. And as you can see, they display considerable variability. However, as shown on the figure on the right, median BMFs are higher than 1 for compounds with most frequently reported, namely PFAS and Phos, which suggests biomagnification. The overall trend is an increase of BMF with increasing chain length in both carboxylates and sulfonates homologous series. Sources of BMF variation include uncertainty, associated, for instance, to study design, and variability, including feeding ecology, life traits, and so on. In addition, BMF assumptions may not always be met in ecosystems where steady-state conditions are not guaranteed. A complementary approach in weight of evidence by accumulation assessment could rely on the elimination half-life estimation, which is correlated with BMF. And finally, it should be noted that BMF estimates for terrestrial ecosystem are relatively scarce. There are fewer data for TMF than for BMFs, likely because TMF determination is much more labor-intensive. As shown here, TMF display lower variability than BMF, but they are still variable across studies, with the max-mean ratio ranging between 4 and 38, depending on the compound. Median TMF are usually close to or higher than 1 for frequently reported PFAS, but they remain relatively low, lower than 3. And no clear trend was observed within the carboxylate homologous series. Overall, no trend was observed for TMFs across aquatic ecosystem types. However, higher TMFs were usually reported for trophic webs, including air-breathing organisms, such as mammals, in relation with respiratory elimination efficiency. Numerous sources of variability can be listed, like sampling design, selection of species or analyzed tissues, data acquisition or treatment, varying contamination levels and patterns, life history traits, and so forth. Besides experimental approaches, modeling can also be implemented to further understand and predict PFAS accumulation in biota. While numerous accumulation models have been developed for neutral lipophilic contaminants, such as PCBs, few models are available for PFAS. Key elements were recently reviewed by De Silva and colleagues. Good quality data on the partitioning properties in biological media and toxicokinetics of neutral compounds were key in the success of modeling approaches. Data gaps were identified, and comparatively, there are fewer data for PFAS. However, using the available ones, several types of models have been implemented for PFAS, and I'll give you a few examples thereafter. Armitage and colleagues developed a mechanistic bioconcentration model for ionogenic organic chemicals, including PFAS. Their model relied on several distribution ratios, such as octanol water, membrane water, and protein water. And as you can see from this figure, model performance was satisfactory as regards whole body burden predictions. Model output for carboxylates are indicated by the open circles connected using different lines. And empirical data for carboxylates are represented by the closed circles surrounded by dashed circle. The model was sensitive to input parameters, and the approach highlighted the need for additional data as regards partitioning. Physiologically based toxicokinetic models for predicting the bioaccumulation of PFAS are still in development phases. PBTK modeling has been widely used as a modeling approach in various contexts, including risk assessment of chemicals. Here I show you the example of a model developed for P4A accumulation in zebrafish, which considered protein binding. It successfully predicted the time profile of P4A in fish liver, and such models are useful for investigating the influence of membrane transporters and highlighting challenges in equilibrium modeling, such as dose dependence of elimination constant rates. Note that the metabolization of precursors may be included in such models. Another group proposed the coupling of a dynamic energy budget model with a one-compartment toxicokinetic model to consider longer timescales and more complex situations. This model was used to predict the PFAS concentrations via the diet during the 2-year-long estuarine life stages of a flatfish, the common sole. Juvenile sole growth and contamination were sensitive to environmental factors, and diet composition proved to be the major influencing variable. In an attempt to consider biotransformation, Glaser and colleagues recently developed a model that incorporates toxicokinetics and bioenergetic components within a food web model. This model simulated the impact of PFOS precursor biotransformation on PFOS apparent biomagnification, and they found that biotransformation rates exceeded precursor gill and fecal loss rates, and therefore likely control PFOS precursor concentration in fish. The model was also used to investigate the implication of PFOS precursors occurrence on other issues, including the ecosystem response time. The last example I'd like to show has not been released yet in the scientific literature, but it's been presented in a recent report by Gobas and colleagues. It is based on activity, which is the concentration of a chemical divided by its solubility in a given environmental media. It was applied at contamination hotspots, and it relied on toxicokinetics and a series of distribution coefficients. The model outputs suggested that steady-state activities of PFOS in the majority of organisms in the aquatic food web were equivalent to those in surface waters, indicating equilibrium and the absence of biomagnification. The model, however, suggested that PFAS would exhibit higher biomagnification in air-breathing organisms. But once again, it appeared that model outputs were highly sensitive to input parameters. So to summarize, PFAS, and in particular PFAS, are ubiquitous in aquatic biota, not only near contaminated sites, but also at remote locations. Their accumulation is considered to be controlled by interaction with phospholipids and different types of proteins, leading to a combination of passive diffusion and active transport processes. The accumulation potential of PFAS in aquatic biota is related to their structural features, like chain length or functional group. PFAS exhibits specific tissue distribution compared to hydrophobic organohalogens, higher levels in blood and liver rather than in adipose tissues. Long-chain carboxylates long-chain PFAS are generally considered as bioaccumulative, and they may biomagnify in aquatic ecosystems, especially when air-breathing organisms are considered. And reactive PFAS may undergo in vivo biotransformation. Data for alternative PFAS of emerging interest are still scarce, and most studies have addressed the issue of bioaccumulation in aquatic ecosystems. While our understanding of PFAS accumulation has made considerable progress over the last decade, a number of data gaps can be identified. Data are needed to further investigate the interaction of wide diversity of PFAS with biological components. Empirical data are also needed for emerging PFAS on their occurrence, tissue distribution, toxicokinetics, and equilibrium partitioning through a combination of lab and field studies. The mechanism responsible for the concentration dependency of PFAS accumulation should be further investigated, ideally under controlled conditions to reduce variability issues and through modeling. Bioconcentration studies at the base of the trophic webs are lacking, although they are essential to parameterize food web models. Similarly, PFAS transfer from sediment to sediment-dwelling biota or paraphytic biofilm is currently less documented and should be further considered. There's also a data gap on biotransformation rates and products of reactive PFAS precursors across taxa, and additional bioaccumulation biomagnification studies for terrestrial biota and food webs are needed. I'll conclude my talk with a list of current challenges and future research suggestions. The reduction of uncertainty, this could include the reduction of uncertainties in PFAS partitioning in biological compartments, which appears a major challenge that could be addressed through a combination of experimental in vitro and in silico approaches, either mechanistic or empirical. This would support modeling and assessment. This would also include the implementation of mechanistic models for both legacy and emerging PFAS to predict accumulation and biomagnification at different timescales. This could also include refining accumulation metrics to cover a wide range of PFAS chemistries based on improved knowledge and understanding of PFAS toxicokinetics. We could also further develop complementary approaches, target analysis, HRMS, top assay, EOF to better estimate the occurrence and contribution of precursors to PFAS accumulation. And I think we should also develop biomagnification model to predict PFAS trophic magnification. That should take into consideration factors that may bias BMF or TMF determination, including biotransformation. With this, I finished this presentation. I'd like once again to thank the organizers of this session for inviting me, and I'd like to thank you for your attention. Thank you.